specifically for December.
Yes.
Okay, let's start. Good. So thank you for coming. It's a pleasure that there's people from outside. My name is Jan van der Meer. So I'm working in Lausanne. Today's actually my last day here. Leaving tomorrow. Sorry, but such is researcher's life. So I'm going to tell a few stories about our research on bioreporters. There's many different aspects of it, and we're taking a look at different things in our research. So I don't want to tell you everything, but I want to focus on a few things which we find fascinating right now. So just to refresh your memory, or for those who have no clue what is
Who have no memory.
I have no memory anymore yet of what are bioreporters. So there's various definitions, but in essence, what most people talk about think about when they talk about bioreporters are engineered organisms. So engineered bacteria, but could also be yeast, could also be human cell lines, in which one or more, let's say, reporter genes is placed, because the idea is that this reporter gene helps you to interrogate a pathway or a signal that you are interested in that particular organism, right? So the trick is that you fuse this reporter gene in such a way that when it's expressed, that is as closely as possible expressed in the way that the natural signal would be expressed in that organism. And then you can, by looking at the formation of the reporter protein, you can actually look at what that signal in that organism would be doing. But somehow there's like now different paths that are taken, and I will show you some ideas about that. So in essence, that means that we have to design some kind of thingy. Hello. Some kind of thingy that people now call genetic circuits. After the SynBio speak, we now have to call it circuits. So we have to build some piece of DNA that we gather from different components, right? Parts, components, that's what we love now, and stick it together in a way or another that we can put it in the cell, that the cell is happy with it and does what we want. So in its simplest form, such a circuit has to have a number of components, like it has to have some kind of a sensor, and typically the sensor is a protein, is a transcription factor, for example, that can react to a signal that we want to measure. This could be a chemical compound, but there's also lots of things like stress reporters, so they react to whatever intermediate thing that is built inside the cell. And then this sensor or another downstream sensor transducer is then capable of turning on a switch, essentially. So we have to integrate the signal into gene expression, because that's what most reporters do. There's now some trends to actually go away from this gene expression, but that's something else. So the idea with, if you take a sensor that is a transcription factor, then it's sort of obvious, because the transcription factor is also capable to stick to the DNA and modify the activity of the promoter. And when it receives a signal, it will turn the promoter on or will turn the promoter off. And that's essentially what happens. So, and then the idea is that this will start to transcribe the reporter gene. You get the formation of reporter protein that you can measure. And we all love the different colors and systems that exist, the different luciferases that are very sensitive, that exist in different wavelengths as well, different fluorescent proteins that we can use, or more classical enzymes that we can use to have readouts in microscopic assays. So the disadvantage of these reported genes is you can only stick so many in it, you know, so 2, perhaps 3, that's about it. Otherwise, the different colors will start to interfere and you cannot very well distinguish the one signal from another. That's a bit a disadvantage if you really want to interrogate like multiple parts of a pathway. So there's also more fancy things. I will explain you one of those, where you don't rely on the diffusion of a chemical compound inside the cell, but you try to intercept this outside the cell in a quantum-like manner that anticipates on the membrane. Sorry, I've been discussing with Forrest too much. So a membrane receptor that actually intercepts the signal and then transmits the signal to the inside to another transcription factor, and then the whole story starts the same way. So there's many different ways that you can actually look at it. And the field of bioreporters is some 20 years old, and different lines evolve, if you want to see it like this. So it's used as a cheap, simple alternative analytics. That's also how we develop many systems, because we think this is kind of cool. Instead of having your GCMS or so, you can just have your bacteria measure something. And this is one of the things that we're trying to commercialize, a kit for arsenic detection in groundwater that you can use in the fields. That's based on a bacterium that produces luciferase. So it's a closed system. That's very important for regulations. It's a closed vial. The bacteria are freeze-dried, and you can inject the water. You wait a while, and then from the light that is emitted, you can calculate how much arsenic has been in there. So this is very interesting. There's a lot of let's say, programs that ask for biosensor development in the European community, things associated with buoys, with robots, with automated handheld devices, all kinds of stuff, just to have very specific targeted measurements using biology instead of some kind of chemical thing. But then bioreporters are also used under conditions where it is actually relatively difficult to use chemistry to predict what the organisms are seeing. So this is typically this idea of bioavailability. You know, if an organism has a toxic effect, it's exposed to something, then it's not sufficient just to measure the total amount of chemical, because the total amount of chemical is not necessarily representative for the toxic dose. So there people have thought, oh, it's much more easy to actually ask the organism what is the toxic dose that it sees, and then relate that signal back. So that's also why bioreporters are used to actually look at the biological effects on the organism, the effect of mixtures, combination of different stresses, and so that you could all integrate. And then again, there is another specific aspect of bioreporters, and that is probably in this community program that we are working on here. That is, you can label your organisms inside a community and try to figure out what are the bacteria doing in this particular community, what is the variability, and then you get into the area of microscale measurements, microscale conditions, like what is a bacterium experiencing when it sits on a plant leaf. This is rather difficult to measure by GCMS, but the bacterium that sits on the leaf, you could make a reporter that tells you how much sugar that bacterial cell is seeing that leaks from the leaf and so on. That's otherwise rather impossible to measure. So there's different ways that people use it, and you can probably use your imagination to do other ways. There's many people in synthetic biology now that like just to play with the gene circuits and see, okay, if you turn this component around, what's happened? Can you make transitions, oscillations? For the practical applications, there's not much yet in that sense that improves what has been done before, but it's just interesting to see, okay, how do pieces of DNA fit together? What's the effect? Okay, so typically bioreports are, as I told you, are applied in different formats. So the most common one is in liquid. And in liquid, the idea is that your cells, they are in suspension, or they are in could be a water droplet, could be in the ocean, could be anything. And that by diffusion, the target chemical reaches the cell. So the bacteria are not actively trying to take this stuff in, but they are dependent on, in most cases, on diffusion, bombardment of molecules to the surface. Some of them go inside. That's what elicits then the signal, and then you wait for a while until you see your reporter signal. Okay, but there's also other. We have experimented with the bacterial sniffers, so that will sniff volatile molecules. You know, many molecules are volatile, so in some cases it's not really practical to work in liquid phase, but it's much more easy if you have solid phase things where you want to see if there's something in there, just to rely on gas diffusion, because the diffusion rate in the gas phase is much faster and you can actually measure better than in the liquid phase. So in that case, your bacteria or cells are immobilized in a small droplet, and you get a partitioning through the gas phase of compounds that you want to detect. And then there's things like diffusive gels, whatever, and there's people that have worked with foodstuffs and with urine, and you can, I mean, you can name it, whatever you like, you can try to develop. So the key of quantification is very similar to what you do with HPLC or GCMS, is that you run a number of standard concentrations, which you incubate with your assays, with your bacteria or with your yeast or with your human cell lines. And then you get a particular readout. Here, this is a readout in a light signal that you can measure. This gives you sort of a calibration curve. If you have an unknown compound that you want to measure, you measure it at the same time under the same conditions, and you interpolate that light signal then onto your calibration curve, and that gives you then the equivalent concentration of your standards. So that is pretty straightforward, although there's a lot of discussion, like in this case, for example, if you measure arsenic, people that know the geochemistry of arsenic, it's difficult because you have arsenic 3, arsenic 5, they react differently, you have methylated and so on. So it could be that in an unknown sample, you measure a reaction that you can relate to an equivalent concentration of arsenite in your calibration, but the sample could be composed of arsenic 5, arsenic 3, methylated arsenic, and so on, that combined give you this equivalent effect, okay? So that's what chemists don't like. They will say, we want to know how much is of one and the other and not a combined mixture. So however you see it, it has its advantages or disadvantages. But that's the basis for the quantification. We are not there. I think nobody's there that can say we can measure an absolute amount of, let's say, GFP molecules in the cell. We cannot measure 10,500 molecules and say this is the absolute amount. It would be good if you could, but it's rather difficult and not done very well. So I'll tell you 3 stories about what we're doing in the lab. One of them is very important because we're interested in the idea of having bioreporters as analytical devices. So basically what others and what we have done is exploit nature. Like if you want to build a mercury detector, well, you go and find a bacterium that has mercury resistance, because if it has mercury resistance, then it has a transcription factor that detects mercury and so on and so forth, okay? But if now tomorrow I'm sick and I need to take antibiotics and then somebody comes and tells me, can you make a sensor for ciprofloxacin? Then I go like, well, perhaps, but it would take you like perhaps half a year or a year first to find an organism that reacts to it. There's perhaps a transcription factor. So there have been some ideas, if it's possible by protein design, actually to design transcription factor sensor-like molecules that would then do the job. I will tell you a story about it. And then the second story I would like to present you is actually how you can capture information from individual cells, like in a community or in a complex system, and then some, show you some crazy ideas of things that we think are interesting and that may work or may not work in the end. But anyway, we have the money to do it and so on. So, yeah, well.
Your fund is listening.
Huh?
You're on air.
I hope. It's all very serious and solid science. Okay, so here's the deal. This is a story that frustrated us quite a bit for a long time, actually. But I think it's an important lesson that we have learned, which I hope to convey to you as well. So there has been this idea that a class of proteins that are called periplasmic binding proteins, they're very common in bacteria, so they are like these Pac-Mans that circulate in the periplasm, and the idea is that they can scavenge nutrients for the bacterial cell, and then when they do so, when they scavenge this, they present it to either a chemoreceptor that then interacts with the chemotaxis response, for example, or with a transport channel, like if it's a scarce metal that you have to be very careful about, and it's presented to a transport channel to increase the rate of transport of that thing inside. So there have been people that says, okay, periplasmic binding proteins, we know the crystal structure of that. We can build nice models like is shown here for the ribose binding protein. And then, well, let's just open our trick box of protein design and modeling. And then if now this is ribose that is binding in here, we can redesign it to anything we like, right? We know the crystal structure, just I will let our models run. So some 10 years ago already, there was this group around Hellinga in the United States who made some very impressive publications. And at some point we said, this is great. You know, if that works, then we don't have to worry at all. We can just re-engineer those periplasmic binding proteins, stick it in the computer, and tomorrow we'll have our ciprofloxacin binding molecule. That would be great, right? So one of the predictions that they made, and actually that they tried to demonstrate in that paper, was that you could change this protein from binding ribose to binding TNT. I don't know if you know what's TNT. It's this explosive trinitrotoluene. It's quite a different molecule actually as sugar. You know, sugar has some hydroxy groups, and this is all nitro groups, and it's a tautomeric structure. Oh, there we go again. So we said, this is fascinating. Let's do it. So we said, but before we're actually going to develop something, let's try and repeat what they have done. This was something we should never have done, but anyway, we set out to repeat it. So it's a pretty complicated sensor to make, because what you have to do first is, it's not natural that ribose binding protein actually elicits gene expression, okay? So the trick to do is that people already for a long time knew that you can make hybrid membrane receptors that on the one hand contain the chemoreceptor domain of chemotaxis, that are fused by magic via a protein part that's called the HAMP linker. It's a very mysterious part actually, and accidentally this worked, because if you try to do it rationally, it doesn't work. Accidentally it worked. To fuse that with the cytoplasmic domain of the osmolarity response system NZ, okay? So this protein was called TRZI. You can express it in E. coli, and then when ribose binding protein binds ribose, then it elicits a phosphorylation on the cytoplasmic part of NZ, and this phosphorylation is then relayed to OMP-R, and phosphorylated OMP-R specifically binds the OMP-C promoter, and if you hook that to GFP, then you get a nice signal. I will show you. So you need several components. You need the ribose binding protein, you need the OMP-R, you need TRZI, and so on. So it's a bit complicated sensor. So we said, okay, the sequence is known, it's published, all the mutations that are needed to go from ribose to TNT. You see, it's quite a different molecule, you know, in structure and in chemistry. It required some 9 mutations, but nowadays that's not a big deal. You just type it in on DNA 2.0, you wait 2 weeks and you get back your DNA, so you don't have to make it yourself. You clone it in, and then it should work, right? Unfortunately, it didn't. It didn't to such an extent that it was really worrying. And then the lesson that we learned is, so it's very, very hard to reproduce a negative, or to confirm a negative result, because you start worrying that, well, there's something else that's not okay, right? So this was the first result we got. We built the sensor, the wild type sensor for ribose, which actually works pretty well. So here's the output of fluorescence and ligand concentrations. It's pretty sensitive. If you think about it, but the TNT one, nothing. Well, we said, okay, is the protein perhaps not produced? So we looked at it, the protein is produced in E. coli, we purified it, we purified RBSB, RBSB nicely binds ribose, TNT, nothing, it's just the protein, it's not doing anything at all. So the end of the story is that we could, since there's now journals that liked when you reproduce so you can publish that, and I think it's very important, because this was 10 years and many people have tried this and it doesn't work. There is something in there that's just either nonsense or an accidental mistake that we don't know. Anyway, so people who want to try it have to be careful that you cannot just go from one step to the other, probably because the protein doesn't like this folding, and I don't know what data they have produced, but it's not there. So we are still working on that in that sense. Oh yeah, so these are the different mutations, so 9 mutations. And we said the concept would be fantastic, so we have to go further on this, but it's a more hardcore biochemistry. So one of the things that we do is we do an alanine scanning, so we change all the positions in the protein for an alanine and look at the effect on the ribose binding and signaling. So we find the number of them that cause a loss of function. And some of those, interestingly, were the ones that were mutated in this TNT binding protein. So that already tells us, hey, there's something really fishy in there. And then try to rebuild the modeling, perhaps on this information, to say, okay, this you cannot touch, and there you have to be careful, and so on. But it's a very, very long and tedious work. And the problem is you are very unsecure because you think you're doing something wrong, right? And it's actually contamination in the literature. So that told us to be extremely careful about things. Good. The next story is completely different, right? It's what you could do, for example, with bioreporters. I talked already about this problem that we are interested in. One of the things in the lab that we're interested in is to see, can you seed existing ecosystems with a bacterium that you're interested in? For example, to add a functionality to the ecosystem that's not currently there. And the functionalities we like to add is degradation of substances which are contaminants, which are piled up in soils and which are not going away. And we know there are bacteria that can degrade it, so one of the goals is actually to put them in, stick them in, and then hope that they can do it. But hoping is not enough. You need to actually look at what's going on. So one of the things that I have told you, or some of you already about, is the transcriptome and so on. But we also work with gene reporters with the idea that, okay, you can introduce your cells in microcosms, they will go, they will grow, and then you can get them back out, interrogate, like, what have they seen? How did they react? Unfortunately, soil is one of the things that you cannot easily plug under your confocal microscope because of the particles, so you cannot actually see very well. So you have to, for now at least, do something where you can put them in and then you have to wash them out and then see how they reacted. But I just want to present this story here as the type of techniques that we use, at least to, for example, address the question, so what's the local availability of the substrate in the soil? Because this is a very big thing that people will say, okay, if you have a contaminated site, the stuff is there, but it's just not available to the cell. It's either too toxic or it lays there as chunks. They cannot get access to it. It's not a liquid culture that you shake. So in this case, it was extremely difficult because the organism we wanted to work with, Sphingomonas, there was no genetic system. This is a very crazy organism. It likes to take up linear DNA and sticks it somewhere, but not where we wanted to have it. So it took us a while to actually come up with a system that reasonably produced fluorescence, as shown here. So this is liquid culture of cultures growing on this aromatic compound that's called dibenzofurane. So we said, okay, at this point, yeah, we can work with it. And this is the type of calibration that you can do. So you can run multiple flasks, measure the average fluorescence, and then plot it. And then you see, oh, when they grow with this substance, yes, the signal is more induced than if they grow on salicylate or another aromatic compound. So there is really an activity of the cells towards this compound, and it can tell us when they are seeing it, it would have to show up as an increased fluorescent signal. If they are not seeing it, then the signal is much lower. But obviously, you can see that already here, it's a distribution, right? Not all cells react to the same way, so it's some kind of a distribution. If you look at single cells, you're starting to see this distribution. If you stick it in a fluorimeter, you don't see the distribution, then you only see like the standard deviation of triplicates, but you don't see the distribution. But it's actually the distribution that becomes extremely important when you go to complex systems like a soil or a plant leaf or perhaps the gut if you want to do this. So to give you some of the data, so what we looked at here, for example, is when we when we inoculate this strain into soil, so this is survival of this strain, and this is then the expression of such a marker that we get, so the average expression of such markers, right? So I'll guide you through it. So this is the same graph, but this is linear and this is a log plot. So if we put cells in sand that we contaminate artificially ourselves with the substrate that they like, the dibenzofurane or the DBF, you can see that they grow. So this is red spots. So they grow over 4 or 5 days, and then they decline because they have eaten all that there is. However, if you put them just in soil, so this is this sand, this is just the soil alone, you can see that they don't do much, right? So we can see on the log plot they grow a little bit, so they have 2-3 cell divisions, but then they go away. Okay, so why is that? What are they doing? You can't tell from this plot, right? Then we also did an experiment where we mixed the sand with the dibenzofurane and this J, that's a material that comes from Geneva. It was a contaminated site, a gasification work that has lots of aromatic compounds. So we said, it's interesting, that's like a real stuff. So we mix it with 10% in the sand because otherwise it was too toxic, and then we looked at what happens. So the cells can still grow to some extent, but they disappear earlier than what's happening here. And then if you just use that material alone, then the cells don't do much at all. That's this blue line. You can see on the log plot that they still grow a little bit, but then they sort of stop. Now, if you look at the marker of this pathway, so what are the cells doing? If you give them the substrate they like, bang, so they induce it, all cells seem to be happy, right? This is the average of the signal in triplicates with the confidence interval, so they're pretty happy. However, if you mix this other contaminated material underneath, then you can see that they do a little bit, but then they They don't really develop very much, whereas here they can grow to some extent. So why is this difference? We don't know. And then if you have only the contaminated material, they still do a little bit, and here's the sand alone, they don't do anything at all anymore. But that's averages, right? So now can we also look at distributions? Because in distributions you actually have, this is the kind of images that you get, right? They're in the sand particles, so if you look in phase contrast, you'll have a hard time seeing which cells they are, and there's other bacteria as well in there. But if you look at the GFP, then you get like these kind of things. Okay, so this is the data that we then interpret. We make lots of images, extract quantitatively what the fluorescent signal is, and then make the distribution. So these are, for example, smoothened histograms of the fluorescent signal. So this is all scaled to the same fluorescent signal over time in the sand. So if you look just at this thing where they grow very well, the sand with the substrate that they like, then shortly after you inoculate them, you see that there's a subpopulation. So there's cells that come in, perhaps they come in at a place where they can't find too much, and then there's some that come in places where there's actually more substrate. This is an insoluble substrate, I have to tell you, so it's not just sugar that you dissolve. It has a solubility of 5 milligrams per liter, so it sticks its patches on the soil. Right? But then after 5 days, you can see that the fluorescence shifted to a much higher level, and almost all cells are now active. So that could be those cells that have started to multiply everywhere, and the others have just declined. Now, if you mix in this other contaminated material, you see that at day 1, they sort of still do the same, but you see at day 5, they can't. They haven't found.
Might they have moved?
Huh?
Maybe they moved?
They can move. They can move. Yes, they can. Yeah. But the point I'm trying to make here is that you can see from the distribution curves that the whole population cannot react in the same way as when all of them see the substrate. But still, if we look at this part, and I'll show you another graph of that in a minute, you can see that some cells are still seem to see patches of something that they like. And even in the sand with the contaminated material alone, so not this substrate that they like, you see that the distribution curve tells you there are still some cells that are in patches where they see the substrate that they like. And that's perhaps more easily appreciated by this kind of plots. So this is a type of plotting that we developed to look at subpopulations, because it's really very hard actually from distributions like this to judge like, okay, yes, it's different between this one, but what is the difference actually? So in one way of finding subpopulations is to plot it as so-called quantile plots. So you plot the expected normal distribution versus the real distribution of the fluorescence. And if everything is normally distributed, you will get a straight line. So if there's some cells like here that deviate significantly from such a straight line, that tells you then that there is actually a subpopulation of cells that behaves completely different. And then you can quantify this subpopulation. So in this case, it's 5%, but it's actually only 2, 4, 6 cells. So you have to be a bit careful about what you're saying here. But these cells have a much higher fluorescence than you would expect from the normal distribution, right? So these things are helpful to say, like, okay, in this case, for example, we still see some cells that find something to eat, but the majority of cells cannot establish itself. So that helps us then to understand why, under these conditions, for example, these cells that you introduce cannot establish themselves because they cannot find the food that they are looking for. But still, some of them are active, right? Good. Okay. You can ask questions as we go along, because now we're starting for some more of the crazier IDs, so application type of things. So, yeah, sure.
Yeah, sorry. Just about the conditions of the experiment that you were describing now. So you take soil samples and then I think you suspend it in water or media.
So what we do, these are experiments with sand that we moisten with the medium that we put the cells in. So the cells have also salts and nitrogen and that kind of stuff and so on.
Okay.
And then we mix in either, let's go back. Then, for example, we mix in either this contaminated material at 10%, just by physical mixing, and then keep the moisture always at 10% total moisture in the system. Yeah.
And you mentioned that there are other bacteria in the sample.
Yes, there are.
So you're not using selective pressure, no antibiotics?
No, nothing. No.
Okay.
So we also know that so we can let this run for 8 days, or we can let this run like for the 7 or 8 generations of growth. But unfortunately, this strain will lose the reporter afterwards. So we cannot re-inoculate from here into the next batch without antibiotic pressure then.
Because my concern is that you're getting co-cultures with unknown organisms. I don't know if you've characterized them, and co-cultures do affect the behavior of systems, especially if
Exactly.
since they came from an area that's affected by these things.
Definitely.
They reduce them and oxidate them to a state that can be toxic to other cells as a competitive
Exactly.
advantage.
Yeah. So one of the ideas that we have to explain this kind of behavior here is that actually these other cells do not compete for the primary substrate, but they compete for a metabolite that these cells are excreting during the degradation of this compound, and it's the metabolites that the others are feeding on. So they sort of lose part of their of their food that they are producing themselves. Yeah. But it was sort of interesting to see that. Okay, so some of the other things that we are doing in terms of applications. So one of the things that we really like is to build miniature chemostats. Perhaps Christian will tell you again what's a chemostat, but I'm sure that most of you will know what sort of a chemostat. The purists, they will tell me that what we have here is not a chemostat because it's not continuous, it's semi-continuous. So what it is actually, this is a 2-layered PDMS chip. You can see some of the structures in here. On there is a growth chamber below. So these have a height of 50 microns, and here there's like a millimeter wide. And they're separated from the medium by these kind of things. So very, very shallow channels that only have a height of 500 nanometers, so that cells in principle cannot escape through and go to the medium. Obviously they will in the end because the bacteria are cleverer than we, because they can change their size. So what you can see here, for example, is this sort of semi-continuous movement, because what we do is we have valves here. So this is basically a plastic material, so you push with air or with water on top of the channels to push them down, and then the thing is closed, so you can open it and then you get medium flow in, and then you quickly push them down again. So you open these zones and then they get fresh medium, and then you close them again to let them grow. So to have this sort of semi-continuous thing, you give them new medium, they divide, and then you push a part of the cells out again, so you get this sort of dilution rate in this miniature chemostat. You can see that a little bit in this movie that's running here. So every once in a while you see the medium coming in and then they push the cells backwards. So these are the cells. You see, there they push backwards, but then immediately they go back to where the medium has come from, because that's what they most sense. So this is.
What frequency do you do this?
Yeah, so on top of my head, we do it every couple of minutes for 10 milliseconds or so. So it's relatively short, because otherwise you wash them out. So that's why purists don't call it a real chemostat, because in a real chemostat you have a vessel, it's mixed, right? So here we don't have mixing. And then you drip in new medium, and every drip that you drip in, you remove the same volume on the other side. Okay?
But dripping also has frequency.
Yeah, but the volume of the drop compared to the volume of the reactor is much smaller than these volumes here, which is why we cannot work with volumes, but we have to work with opening and closing. Also because these systems, they are driven by pressure, they're not driven by flow. Flow is very difficult in these things. Okay, so our idea was they can happily grow here, and then whenever we would like to make a measurement, this is the idea that, for example, on a buoy, in a marine zone, in a harbor or so, you could have these chips running with your bioreporter bacteria all the time. So as you know, a chemostat produces waste because the cells are growing, and every time you open here, you push some cells to the outside. So the idea was that we could collect those cells actually here in a measurement zone that looks like this, or like this. If you can see that cells are accumulating here, because also here there's these pores that stop them. And then you can push the sample through, and then you can watch the glow coming from the cells continuous in semi-real time or something. That was the fun of it. Obviously this thing breaks down very easily because it's not so robust and so on, but I think the concept could be interesting, because one of the limiting things in applying bioreporters is if you have no one if you do not have a lab where somebody pipettes something, then it's really hard, you know, to have an automatic system that takes a sample, brings it to your cells. The cells have to be active all the time, otherwise they won't react. So this is one of our attempts to do this. So the second project is actually this. This is even more crazy. This is a robot. It's a snail. So this robot is built of segments that you see here. So the segments are about this size. So it was a robot built by the people at the EPFL robotic laboratory. So this thing can swim. So it has this kind of anguilliform movement, goes back and forth. But when we saw this, this is cool when you see those pictures. But what happens is, and there's a guy standing on the side using a joystick to pilot that thing, right? Because they're interested in how do you program this movement, and how does that thing make a curve, and why is it not sinking and going up and so. But we said, no, come on, you can do better than this. No, we have to do something that this thing can find its own way. That would be cool, no? That you have something in the head of the robot or in one of those components that actually tells the robot, okay, I'm seeing here that there is an oil spill, let's go to the right, so that it can actually track. This is a very complicated computational program as well, because if you would have measurements, then this thing would have to integrate on the way these measurements to understand where am I going, and that's a very difficult problem, apparently, for computation biologists. Anyway, so the whole program is around how can we put sensors in those kind of modules. And this is obviously a very challenging problem, because, I don't know if you noticed, but most of the bioreporters are rather slow. So if you wait for gene expression, this takes, well, I mean, we know a gene is expressed in 5 minutes, but to see a reporter protein takes half an hour or an hour and so on. That's not really very practical if you have a thing moving through the water, right? Because then it would have to sit idle for an hour until it gets its measurement and then decide, okay, where am I going next? And by that time, so this is one of the big things. For now, we're trying to argue our way around that it would still be interesting to have this thing going on a mission, let's say a mission, and you send it into the river and say, take a sample there. Okay, so it will go there, takes a sample, and wait for a while, and then go back in. So the things that, for example, we are envisioning is this. So this is a very small flow cell that you can fit in here, where you have electrical connection, so you need an electrochemical sensor, but you can put bioreporters here, and they are blocked by magnets that let the flow in and then stop the flow. And you can capture the signal from the cells also by using an electrochemical active substrate, and that produces then electricity that actually then is captured here, and that directs the movement. But another thing that we are sort of in the long term are working at is, could we use chemotaxis? Could we use bacterial chemotaxis? Right, you know, bacteria can also swim, and the idea is that they can also measure gradients temporarily and decide actually where to go. So we said, is it possible to make a quantitative readout of chemotaxis? And this, as it turns out, is also one of these things. The literature is full of chemotaxis, but when you're trying to measure it, nobody knows how, and it's extremely difficult, we find. Perhaps we're doing everything wrong or so. So the thing that we are developing for now is again a microfluidic cell, which has 3 lines, as shown here. And these lines are again separated by these very thin channels, so that bacteria that are in the middle channel, they cannot escape to the outside. And the idea is, if you have 2 parallel channels, one with a buffer, so nothing in there, a sink, and another, we say the attractant, but that could also be the sample, then you will get a gradient form parallel to the channel. Because you have a constant sink and you have a source, then you will get a gradient perpendicular to the channel, okay? That would mean that, for example, in the case of attractant, that the concentration of substance would be higher on this side and lower on that side. And if the bacteria are capable of reacting to this, then they would move preferentially to one side because they see that the gradient is there, and they would go distant from the other side, okay? So we put bacteria on the inside, and here, if you can see properly, it's a mixture of 2 E. coli, green and red, like here. Red is a mutant that we use as an internal control. It's a mutant in a flagella, so it cannot swim. I mean, it can go a little bit here and there and go with the flow, but it cannot swim. Whereas the green is a wild type E. coli, so it can swim. So this is, for now, one of the few experiments that worked, which is why I'm showing it. So it's actually using a serine gradient, a steering gradient from high to low, and the bacteria being pushed in the chamber. But the trick is you have to have a very, very slow flow of bacterial cells in this chamber, and a very, very slow flow of both components on the side. Otherwise, there is so much flow coming into the chamber that this takes all your bacteria and they cannot swim anymore. So what you can see here
How slow is very slow?
I should be able to tell you, but the problem in microfluidics is, do you want to have the flow in cubic volume per second, or do you want to have it in
10 microns per second?
No.
1 millimeter per second?
No, not even. So this must be in the order of 1 micrometer per second. Okay, so and these are flow lines, again, with a height of 50 microns. So it's very, very small flows, because otherwise you push the cells away and you cannot see the chemotaxis. And then the idea is that you can look across, so you can do a perpendicular scan of where the cells are, and this picture is obviously a little self-illustrative, that you see that they accumulate, in this case preferentially on this side, and there's very few on the side where there's nothing to eat. And if you quantify, then you can see that you can actually quantify that there's many, many more on this side than that. Now, how to translate this into a system? So this is pretty rapid. So this is like 3 minutes, 5 minutes. You'll see that the cells start to move to one of those directions already. So you could imagine that such a system, if you replace the fluorescence by an electrical detector or so, that you could actually get the signals from the cells if they see something that they like, or if they don't. If they see something that they don't like, they would move to the other side. But that's the type of things that we hope to achieve there. Okay, final piece. Sorry, it's so fragmentary, but just to give you some idea of what we do, was that you could also use bioreporters in combination with something else, like other living beings. And we were fascinated for a long time already with nematodes. So this is C. elegans nematode. No, it's actually not. This is from a garden, so what must be some other nematode. Okay. And some ecotoxicologists, they told us, like, okay, if you have a soil or so, there's abundant quantities of nematodes. That must be like dozens per gram or hundreds per gram, which are apparently relatively easy to isolate. So we said, well, would it be possible that you can actually have bacterial bioreporters inside the gut of the C. elegans? That could be interesting for various reasons. Could also be interesting for medical reasons, but unfortunately we are not interested so much in medical reasons, but only environmental reasons. But we said, okay, if a nematode, imagine it going to the soil or so, and this soil is contaminated, then some of the stuff that it eats will be contaminated, right? Probably then some of the contaminated stuff will be in its gut, because that's what it's eating all the time. And then if we could use the bioreporters in the gut to measure how much of the contaminant is in the gut, then we could use the nematodes as a sensor and then apply the bacteria and then get the readout, right? So the good thing is nematodes, they like bacteria. Actually, they eat bacteria, so you feed them on bacteria. This is also how you can isolate them. So you can put a soil sample on a plate and then have some bacteria, and if they like those bacteria, they will crawl towards it, start eating them. And if these are reporter bacteria, then you can get a signal of the bacteria inside the gut. So very young nematodes, they crunch the bacteria, so there's nothing left. But older adults, they are not crunching the bacteria so efficiently anymore, and then the bacteria can actually survive in the gut. And you can also, you know, like, I don't know how you call it, you can also sort of block these pharynx with chemical tricks so that they eat but they don't crunch, and so that the bacteria end up alive and not dead in there. Otherwise, yeah, your signal will be sort of gone. So what we have here is an E. coli sensor for arsenic that produces 2 colors to give us an internal reference, so a color that's always produced, and then another color in green here that tells us, okay, if there's lots of the stuff, then more green and so on. So that's why we plot here a ratio between the green and the red. If the ratio is higher, then the cells have seen more of this stuff. So here is, for example, nematodes that we get just from garden soil, and then we get a very, very low signal, so there's not a lot of induction here. But here's nematodes that we picked up from a soil contaminated with arsenic, And then you can see that actually the cell, the bacteria inside the nematode gut, they respond much more wildly to the substance, so indicating that something is in that gut that elicits the signal in the bioreporters. So we think this is sort of a strange twist to the story, but what it tells me is that there's a lot of possible applications that you can actually do for bioreporters if you think about it and then work with it. Good. So that's about what I wanted to say. If you're interested, so there's a number of European projects. I like to make some, advertisements. So we have this Bravo project. We probably got it because of the name, where we are supposed to develop marine buoys that are operated with sensors. This is very, very challenging. And then there's this EnviroBot project with the snake, which is also very, very interesting. So thank you very much. I hope it wasn't too fragmentary, but I'll be happy to answer questions. Yes?
So what are you doing on the marine buoys?
What are we doing on the
What
marine?
what can you measure on marine buoys?
Well, the idea with the marine buoys is that we measure compounds that are important from a perspective of aquaculture. And that is primarily, so yeah, I should talk to you about cyanobacterial toxins, because those toxins is one of the things that people really, really like to measure very well. And this is extremely difficult. And the other thing are antibiotics, for example, which are widely used in fish farms.
With reporter bacteria?
Yes, among others. But this project is a multidisciplinary project, so we have 3 types of sensor groups. So there's people that work with immunosensors. So there's certain things that you can detect with the immunosensors because you have antibodies against certain compounds, so that makes it easier. Then there is a group working with actually photosynthetic algae to use the photosystem as a readout for toxicity. And then there's 2 bacterial biosensor groups where we try to build, let's say, specific targeted sensors. But the big problem is not so much, everybody, I mean, I can even teach Todd here to make an assay with bacterial reporters in the lab. That's a piece of cake. He can even do the cloning.
You've never seen me try anything.
Well, I imagine, or let's say Daniel, so he could do it. But the challenge in a marine system is that, so people who work there, they will laugh at us because they will say, you come with your microfluidics, that's all nice, but you know that a flow line of, let's say, a millimeter is clocked in half a day in a marine.
Yeah, the fouling on your sensors.
So the fouling on the sensors is considerable, and then the salt.
That you're absent for a month or so.
Exactly, that the boy has to lay there for a month. You don't want to go out with the boat every time. So that is an extreme challenge. So we think there's a number of possibilities there. So there's groups that are so good in making, like single dose type of things that you can think that you have single dose sensors, and you inject the water in it automatically, and then afterwards it's just thrown away. And this can be very small, so we hope that we can find a compromise that will actually fit that in a buoy, let's say. But yes, it's very, very challenging. But on the other hand, there is a lot of interest, like we want to measure everything everywhere, It's and that's very hard, because to miniaturize a GCMS on a platform, wow, that's not an easy thing. And that will clog as well, so. Yeah. Yes?
Could you say a few more words about the antibiotics project with fish? What kind of constructs do you use?
You mean in this marine?
No, you just mentioned that there's a project with fish.
Yeah, so the aqua farming, yeah. Well, the thing with antibiotics is that some sensors are known from E. coli that are sort of unspecific promoters, so they're not related to antibiotic resistance. These are screens that people have done just using libraries of all promoters in E. coli, and then see which one lights up if you give E. coli, whatever, an antibiotic X or so. And then there's a few targeted antibiotic sensors like tetracycline, for example, that people have looked at. But there is now really a renewed interest to be able to not only differentiate antibiotics, but detect them at the low level that you can find them in fish farms and so on, and in surface waters. So it's a very, very big challenge because antibiotics are also very, very difficult to measure chemically. You know, you have to concentrate the stuff, purify it, put it on. It's a lot of steps. And this is I think the primary incentive for many of these biosensor projects. If you have, let's imagine you are you're responsible for the water quality in a harbor, or you run your fish farm or so, and you want to know what is the chemical quality there. What you have to do now is you take a sample, you send it to a lab, you wait 2 weeks, and then you have your result. That's not going to work, right? If you want to do something about it. So you need something that you can reasonably take the sample, and in an hour or so you know the answer, or 2 hours perhaps. And that's what many people are actually looking at, if this is possible. Yes.
So the biological safety aspect of the buoy is released at sea. So I understand you're intending to put GMOs on a buoy in the ocean, leave it there for a month.
Well, it's interesting that I'm not saying that we intend this, but my opinion is that there is extremely little risk. Actually, I mean, there is it's not a non-zero risk, but I think it is comparatively little to other risks. But it's amazing how little this aspect is discussed at the level of the people that ask for these calls. You know, nobody asked us a single question like, okay, what you are trying to do is put GMOs on a buoy in a marine system. You know, we can argue that, okay, a buoy, you can make it closed, you can put your bacteria in a closed system so that unless there is an explosion on the buoy, a major incident or so, this is not going to be released. But yes, there can be accidents, there can be this and so on and so forth. I can tell you an interesting story if I may. Oh, I still have 6 minutes, yeah. So at one point we tested our bioreporters on a ship. It was actually a research vessel from the NIOS. So this was a very interesting thing because we had to argue with the Dutch government. Can we get permission to work with like 1 milliliter E. coli cultures on a ship? So, and in the end, they didn't know how to deal with this because they couldn't decide whether a ship is a lab or whether a ship is a ship. And because it's outside, you know, it's outside the land, where does the jurisdiction of a local council and then of the national government, so they couldn't decide. So in the end, they gave us a 2 weeks temporary permission to do this. But the irony of the thing was this was an expedition to test the effect of oil spills. So we had permission to dump 10 cubic meters of oil in the sea, which is not a lot. I mean, it gives you 2 square kilometers of oil film, which is gone in 2-3 days, but still. But to use 5 milliliter of E. coli cultures, we have to have a closed container. We have to keep book of everything. We had to have autoclaves on board. We had to have everything, you know. Mm-hmm. So that was, it was an interesting experience. Yeah.
But it's the same what you need in a laboratory, right?
Yeah.
If you want to work with different strains
yes.
of E. coli, required
yeah.
have autoclaves. And
so you're able to convert the ship into CL1?
Yeah, well, safety level 1 was okay, but safety level 2 would not have worked.
That's not
no. Yes.
I understand that the biosensors are always like made for one specific component in a way. And I was wondering if there was a chance, I mean, this is some ideas I just had, to sort of label an entire real community with different sorts of biosensors in order to see how the flows of the metabolites are actually going. So I imagine that, there would be some microbes sliding up in the first moment and then others coming up later on and stuff like this.
Yeah.
So this is probably
I think that's possible, but as I'm saying, you get down to the problem, how can you differentiate one or another? So we find that, for example, in using different colors, there's only certain color combinations that we like to use because otherwise the risk is too big that the spectra overlap, and if you have one signal that's high and another one very low, then you start measuring the tail of the high and the low, and then you don't get proper signals anymore. So I think three colors would probably be all right. And if you then can couple it to a completely different reporter, like having fluorescence and bioluminescence at the same time, then you could add a fourth or a fifth probably in there. Yeah.
But this would then be like an artificial community.
Yes.
That type of approach, right?
Yeah, yeah.
There's no way to think some solution for actually getting a real natural community.
Oh yes. There are, I mean, people are working on this now. I mean, at the identification level, people are using fish, for example.
Yeah, of course, but I mean, like, I'm talking about compounds.
Yeah, so this is going into this area of this click chemistry, I think, of which I don't know exactly how it works, but that's the idea that they have behind that, so that with specific chemical, I understand chemical fluorophores, you can label specific proteins in single cells, but I don't know how much pre-treatment it requires and so on and so forth. That's,
Sounds laborious, right?
yeah, and then there's people that obviously do the thing like the Raman scanning, which you can do at microscopic level, in combination with stable isotopes. This can tell you something, and then the nanosims, obviously, but with nanosims, I don't know, do you have experience with nanosims?
Somehow it works.
Yeah, so I see people, you know, doing like a cell here and a cell there, but a whole community.
Exactly. I think the problem is that they only have the max.
Yeah, and you blow away your cell once you
Yes.
analyze it, so it's kind of the quantum microbiology effect. If you interrogate it, it's gone. Yeah. Yeah, I still think that the report is there, so if you do it carefully, then you don't disturb too much the organism that you work with. Yeah.
So why don't we take a 5-minute break?
Perfect. Yeah, thanks a lot.